William Shin. I'm from the Califano Lab at Columbia University, and today I'll be talking a little bit about the work that we've done taking a systems biology approach to the study of Alzheimer's disease. So the Califano Lab in the last few years have developed a number of tools to build and interrogate interactomes. One called RECOMB can be applied to data sets of gene expression profiles to reconstruct a gene regulatory network. And another tool called MARINA, or master regulator analysis, can then ask questions or interrogate this gene regulatory network to infer master regulators. And these are transcription factors whose activities drive the transition from one state to another, such as controlled to diseased. So for my thesis project, I wanted to investigate this question: can these same tools be applied effectively to the study of neurological disorders? Well, moving into this context, we ran into some challenges. The first one had to do with brain tissue heterogeneity. For example, if you take a sample from one region of the brain and another region of the brain, they can exhibit some very different characteristics. And even within a region, you can have multiple cell populations, such as neurons or glia, and this complicates the analysis. Another issue has to do with acquisition of tissue from the site of pathology. It's not as simple as sticking a needle into somebody's head. You can't get the samples very easily, and so it actually poses some challenges in studying the progression of neurological disorders because you can't get those samples very easily. But I think there are some ways to get around these challenges. For instance, the issue of brain tissue heterogeneity, there are some more sophisticated methods of sample collection. An example is a laser capture microdissection, and it's a method that allows for the acquisition of very homogeneous samples from a heterogeneous population. And also, some prior knowledge about the affected regions of that neurological disorder can sort of help us narrow down the regions considered so that we don't overcomplicate our analysis. When it comes to acquiring tissue from the site of pathology, one way we can sort of get around this is by getting samples from people who had intermediate stages of the disease. So these are still post-mortem samples, but it's sort of a way that when we consider the control, late stage, and intermediate samples together, we can sort of get a picture of how the disease progresses. The question is, why Alzheimer's disease? Well, it's the most common form of dementia in the elderly. It affects over 5 million Americans today, and this will increase to over 13 million by mid-century. It's characterized by two hallmarks. First is this deposit of a protein called amyloid beta, and this deposits in the space around the nerve cells. And also, you also find neurofibrillary tangles, and this is what builds up inside the nerve cells and eventually kills them. And of course, these two markers causes the loss of synaptic connections and eventually dementia in people who suffer from this disease. So why study Alzheimer's disease using a systems biology approach? Well, one of the challenges that researchers face in the field is a lack of early diagnostic criteria for the disease, as well as targets for disease-modifying treatment. And our methods allow for the identification of genes whose activities drive AD progression, and hopefully this will allow us to identify new targets, new effective targets for treatment. I'll talk a little bit about the data set that we used in our study. The data set that we used was a public one that was published by Liang et al. In 2009. It's one that contains over 190 samples. And over here, you'll see the different regions that the samples were taken from. On the right, you see the Brock stage. And this is basically a metric that allows you to see, or that represents how far along the disease has progressed. And you see these different regions are progressively affected as the disease gets more severe. And one thing that was really nice about this data set is that it overcame some of the challenges in studying neurological disorders that I talked about. For the method of sample collection, it used laser capture microdissection. So it allowed us to get some very homogeneous samples from these different regions. And also for the issue of disease progression, if you notice up here, there's three groups, so control, AD, and NDAD. NDAD samples are from people who had some earlier stages of the disease. So they had the, had the histopathological markers, but the disease wasn't as progressed enough where they were beginning to show the signs of dementia. So by considering the control, the NDAD, and AD samples, we were hoping to get a better idea of how the disease progresses. Talk a little bit about network reconstruction. So given that data set that I just described, we applied RECOMB to reconstruct a transcriptional network that contained around 500,000 interactions. So the next step was to then interrogate this network to predict the master regulators of AD progression. So here's a little bit of a description of how master regulator analysis works. Well, it begins with what's called a gene expression signature. And essentially, this is a list of genes that are sorted by their differential expression, going from ones that are overexpressed in AD with respect to control on the right, and underexpressed in AD with respect to control on the left in the blue. And what master regulator analysis will do is it'll then overlay the predictions of our interactome. So here's two TFs as an example. And if you look closely at these two TFs, you'll see that TF1, well, all of its activated targets are some of the most overexpressed genes in AD, and all of its repressed targets are some of the most underexpressed genes in AD. We don't see the same thing in TF2, because all of its targets are either not moving at all, or they're moving in the wrong direction. So what master regulator analysis does is it calculates and it predicts things like TF1, whose activities drive this transition. And one thing I do want to mention is that TF1 and TF2 at the mRNA level might not be changing all that much. But really, master regulator analysis looks for genes or TFs who exert their influence through their targets, or the differential expression of their targets. So we took all of our regions, and we took our two comparisons, and we ran master regulator analysis. So these are bar graphs that show the number of master regulator candidates that showed up in each region for our two comparisons, control versus affected and control versus NDAD. As a next step, what we wanted to do is sort of take these lists and prioritize them for further biochemical validation. So in order to do this, we took two strategies. The first one came up when we had a better look at the data set and where the samples were collected. What we noticed is that some samples showed variations that may not be related to AD, confounding factors. Here's two examples of samples from the hippocampus, and they're both affected, so we consider them to be the same, but they were taken from slightly different neurons. They might have a difference in their ethnic background, and of course, there's a different age as well. Our question was, how would this affect our analysis? Because we would want to know which master regulators were consistently highly ranked, regardless of which particular samples were used. So for this, what we did is we applied a bootstrapping step to master regulator analysis, where we took random subsets of each class, and we ran master regulator analysis multiple times. So what bootstrapping told us, well, not all the candidates were consistently highly ranked. And as you might expect, certain candidates were very sensitive to which particular samples were used, and certain other ones were a bit more robust. So they were consistently highly ranked. We called them stable. And for validation, what we wanted to do is focus on the candidates that were stable, and so we filtered them out. And this is what happened when you filter them out. Again, we see in the blue the original list, and in the red is the remaining list after filtering for just the stable master regulator candidates. And certain regions were affected more than others, and we think that we were able to filter out some false positives in this way. Another strategy that we used has to do with the nature of the data set again. Again, we see the different regions that were considered, and you'll notice that they all play some role in the progression of the disease, because they are progressively affected as the disease progresses. And so we reasoned that a master regulator that drives this progression would be enriched in more than one region. So what we did is we went back to that list, and we only considered the ones that were enriched in more than one region. So here's what happens to the list again. So now we have the blue, which was the original one. The red is the ones that were stable according to bootstrapping. And now in the green are master regulators from the red list that show up in more than one region. And from this, we decided to focus on one region in particular, the hippocampus, which has been known to be affected very severely by Alzheimer's disease and has been known to play a role in memory. And so from this, we looked at our highly ranked master regulators. Well, you'll see there's two columns. On the left, we're looking at the green list, and we're sorting it by the metric that's given by master regulator analysis. This is called the odd ratio. And on the right is we're taking that same list, and we're sorting it a little bit differently. You'll see the parentheses on the right, and that's the number that represents the number of regions that this master regulator candidate was enriched in. So TFEB, TFEB was enriched in hippocampus, as well as 4 other regions, for a total of 5. So given the tools that we had available, we picked 4 from these lists that we thought that we could validate effectively biochemically. So for the remainder of my talk, I'll be talking about the 4 that we picked, YY1, EP300, ZMYM3, and MEF2D, which were one of the ones that were enriched in the most areas. So now the biochemical validation. So for this, we collaborated with the Shalansky lab at Columbia University. And they had access to samples from the Columbia University Brain Bank. And again, you'll see that we tried to keep the model consistent. So we looked at control with the moderate AD and severe AD. And since we were comparing this back to our microarray data set, one thing we wanted to do is take these master regulator candidates and compare their gene levels from the whole cell extracts from the brain bank with our microarray data. So that's the first thing we did. And the other thing that we wanted to do is then look at the master regulator candidates at the protein level and see how they change between control, moderate, and severe AD. So I'll be showing that later, but we did immunohistochemistry on tissue microarrays and Western blot analysis. So first, the comparison of the gene levels. So this is what we got from the LCN. So these are the full change of expression between control and AD for our 4 master regulator candidates from our microarray samples. And here are the comparable full change values from the whole tissue extract. We see that for most of them, we see whenever we saw an increase in the microarrays, we see an increase in the whole tissue extract as well. And of course, with MEF2D, it's a little bit preliminary, so I'll be talking a little bit more about EP300, YY1, and ZMYM3. So for ZMYM3, we went down now to the protein level. So this is immunohistochemistry from the CA1 region, which is a region of the hippocampus. And what we're looking for is the brown. So the brown is ZMYM3. And when we look at control and AD, we see there's a clear difference between control and AD for ZMYM3. So there's a clear decrease. So we wanted to quantify this, so we looked at Western blot analysis. Here is the Western blots up here. And you notice that there's multiple bands. And one thing we weren't able to determine at this step was whether or not these bands were the degraded forms of ZMYM3 or if they were different isoforms. So what we did is we considered them together. So down here is the quantification. And the upper doublet is basically these bands up here. And the lower triplet are the 3 Thaner bands down here. But even when we consider them together, we see basically the same trend. That you have a decrease in the amount of ZMYM3 going from control to AD for both of these bands. Next for YY1. For this, it's a little bit more interesting because if you compare control and AD, again, YY1 is in brown, there's not that much of a difference between control and AD. But when we did the Westerns, we found one reason why this was the case. So here are the Westerns for YY1. Each column is a biological replicate. And up here is YY1. And for here, we knew that the lower bands here were the cleave fragments. And you'll notice that there is a bit of variability going from column to column. This kind of is one of the challenges we faced when working with samples from the brain bank, because there are some variabilities with sample preparation, with time of death, with how long it took for the samples to be prepared. But we were still able to see the same trends, that there was a decrease in the full protein and an increase in the degraded fragments going from control to AD. So this is the reason why we weren't seeing it in the immunohistochemistry, because the protein was still there, but it was just getting degraded. So I guess I will go very quickly through this. So this is P300. Again, when we stained for just P300, there wasn't a difference between control and AD. But one thing we knew is that it got phosphorylated in order to be active. And when we stained for the phosphorylated version, we saw a very clear difference between control and AD. And this was nice because if we looked at the gene expression levels, this is not something we would have picked up. But the method looks at the activity, and we were able to pick up the activity of P300. So some of the conclusions, we were able to have an effective approach to the study of Alzheimer's disease. And one thing I wasn't able to talk about is that all these master regulators have some role in histone acetylation. So we suspect that this is one of the things that might be going wrong in AD. So these are some of the people I'd like to thank. And thank you for your attention, and sorry for going over time.
So we're running just a little late, but I think we still have time for a question or two if anyone has a burning question. Well, while our next speaker sets up, if something comes to mind, please step right up. I guess I have a question. To what extent do you think that measurements of the protein levels of these master regulators are sufficient to determine whether there's a change in activity? I mean,
Yeah, so there's a bit of biochemistry that I wasn't able to show. And I think if you go, especially for the P300, we looked at, because even phosphorylation sometimes, right? So we looked at one of the substrates, so P53, and saw how that changed from control to AD. And we found that, indeed, its activity did change between control and AD. And then, so it's not a complete picture, but sort of a beginning step that we can take to check whether or not its activity is changing. Should I give this?
No, if you
I think there might be one question back there, actually.
Quick question. So do you consider also a combination of transcription factors to be a master regulator?
Could you say that one more time?
Do you also consider combinations of transcription factors to be a master regulator? For example, TF1 and TF2, they may not be individually a master regulator, but if you consider, for example, their XOR or some other functions, it can be.
Yes, that is something that master regulator analysis does, is it looks at the synergy. So we look at the targets, and if the targets that they both regulate are significantly more differentially expressed, then we kind of suspect that they might be working together. That's something we did do, but I didn't show in my presentation. Thank you.
Thank you very much. Really great.